Welcome to the Infection Prevention Strategy Tips Masterclass Surface Contamination Detection for Environmental Cleaning. This informative webinar on the critical role of surface contamination detection in enhancing environmental cleaning and disinfection practices. We will explore various surface contamination detection technologies with a focus on surface imaging. Learn how surface imaging technology can revolutionize the way we monitor and remediate contamination, ultimately leading to a safer environment for all of us. The learning objectives for today are posted on the screen for your convenience. My name is Dan Mueller. I'm the Senior Vice President with Corsia Healthcare, and I'm also a TIPS Advisory Board member. The presentation will last 50 minutes, followed by 10 minutes for Q&A. For any questions, you can type your questions into the Q&A panel. The webinar series is hosted by TIPS. TIPS is a not-for-profit that exists to advance innovations, ideas, and processes that make a difference in global health. TIPS extends to over 30 countries and is the key strategic partner for many notable organizations, initiatives, and events. In the past few years, TIPS has contributed millions in scientific impact. Our mission statement is listed on the slide. Join, contribute, make a difference. By participating in this webinar, you acknowledge statements and information provided are for general guidance purposes only and does not replace guidance and direction of your local government and health authority. The webinar series is sponsored by OptiSolve. OptiSolve began as a question which led to an idea. The question was simple: is this surface clean? A small team with an international organization with over 60 years experience in manufacturing cleaning products began working with academics and tech innovators to find solutions that would help cleaning teams identify contaminated surface with far more accuracy. Their Pathfinder and Savvy systems offer visual, evidence-based cleaning validation, aiding in contamination detection, and cleaning effectiveness monitoring. Introducing today's speakers. I'm excited first to introduce Dr. Rodney Rohde. Dr. Rohde is an advisory board member of TIPS and professor, research dean, and chair of the clinical laboratory science program in the College of Health Professionals at Texas State University. He's been recognized with teaching excellence at both Texas State and Austin Community College. He's a frequent keynote speaker traveling across the country. And if I read his CV, we would be here for another half an hour. So Rodney's very recognized and we're happy to have him again. Our next speaker is Mark Kandow. Mark brings over 18 years of expertise in the cleaning and maintenance sector, having held various positions with a prominent distribution firm and cleaning solution manufacturer in Ontario, Canada. His extensive experience has fueled a deep passion for mastering disinfection and infection control. Mark is dedicated to finding and implementing the most effective methods for achieving precision cleaning. And again, if you have any questions, go up to the top of the Zoom screen. You can type your question into the Q&A area. If we can't get to your question, we'll gather and follow up with you later. All right, Rodney.
Good afternoon or good morning, depending on where you're at. And thank you, Dan, for that introduction. It's always an honor and a privilege, really, to be involved with the TIPS Master Series. And I'm really excited today to join OptiSolve in discussing this topic around surface decontamination. So the first set of slides really are going to give you some context around multiple drug-resistant organisms. We're going to jump into a little bit of antimicrobial resistance, as well as biofilm formation. And then I'll introduce some of the technology, and then Mark will take it from there. So many of you know this, but in general, a patient at a healthcare facility with a multiple drug resistant organism, in other words, one single drug, whether it's an antibiotic or an antiviral or even perhaps an antifungal drug, will not work. You need multiple drugs to kind of get that combinational effect. So if you have that type of infection, at least a twofold or more increased risk of death. You're looking at many days of excess length of stay in a hospital and typically in an ICU. That cost is probably way above that, but at least 100K in US dollars. Obviously, you're going to have some patient dissatisfaction. And typically, as we'll mention here going forward, when you have a multiple drug resistant organism, you're really looking at antimicrobial resistance with quite often being integrated with biofilm issues. So here are some of the reasons we worry about surfaces, right? And truly all surfaces matter. And one of the things I like to tell the audience when I'm doing these types of talks is I want you to kind of think about the environment you work in, whether it's healthcare, and we're certainly focused on that today, but it could also be a community setting. And it could really be any facet of healthcare. It could be a typical ICU or hospital setting. It could also be a dialysis clinic or perhaps a surgical unit that's just for day surgery. So it really doesn't matter. All surfaces can harbor and allow certain pathogens to survive on them. And I often like to tell colleagues and friends and family that you really should view a building, surfaces, things associated with those areas as a living environment. And I think you'll understand why when you go forward here. So this is the lineup of many of the common pathogens that survive on surfaces. It is not exhaustive. There are many more, but C. diff is one of the most impactful when you look across these organisms because they create an endospore, which is even more resistant. You might say it is, on steroids. It's so resistant. It can last up to 5 months when you're doing that spore action. And then some other common organisms you might be familiar with, Staphylococcus species, certainly influenza, SARS-CoV-2, which is obviously causes COVID, but another, set of nasty bacterial agents like Pseudomonas and Klebsiella and Acinetobacter, and many, many more, including Candida, which is a kind of an emerging fungal organism. All of those are from, a few hours up to days, up to weeks, and potentially months, depending on cleaning and disinfection of those areas. So it's a bacterial and viral zoo out there within those areas. And when you look at the World Health Organization or you look at other kind of global think tanks around this problem of AMR and biofilm and multiple drug resistant problems, we have a major problem. And in fact, this was long before the COVID-19 pandemic. It's been going for decades. Many of us have been screaming about this for at least a couple of decades. And so this spread has been accelerating quickly. The arsenal for treatment is shrinking, and that R&D pipeline is also very low, or at least very slow as we move forward. There has been some newer antibiotics and some newer drugs pop up in the last 3 to 5 years, but it's still behind the curve. So really prevention and disinfection is the last line of defense. It's all part of a risk reduction strategy. We need a multimodal kind of outlook as we go forward, and surface decontamination is certainly one of them. We also know, if you look at a variety of research studies, and I've actually noticed several coming out in the last 2 weeks, that when someone who has an MDRO or an AMR type of infection and they are in a particular room or even a particular bed within a healthcare setting, you can basically predict that the next person coming in, the next patient, is probably going to have a very high likelihood of acquiring that infection. The World Economic Forum, just to add to this slide, has predicted that by 2050, we will be up to 10 million potential impactful deaths or infections from ANR and an economic impact of $100 trillion. So it is a huge problem globally. So biofilms, right? So there are really 2 basic biofilms when you start thinking about the impact for AMR in healthcare settings. And we've all kind of been following kind of the wet type of biofilms that you kind of think about in a wet environment. But there's also this kind of new challenge that's really just now unfolding within the research setting and the practical setting, and that's called a dry surface biofilm. We'll talk about that a little bit more today. So some basic information around biofilms. Again, many of you may know this, but the context of a biofilm is a living matrix. In other words, biofilms typically are made up of what we call a extracellular polysaccharide matrix, the EPS. And that matrix is really sticky, kind of a jelly-like, and it's microscopic, so you don't typically see it real well. And within that matrix, if you can envision almost like trench warfare, all sorts of bacteria, perhaps fungal agents, other types of agents that are living within tunnels or trenches, like trench warfare. And they're really smart communities because they can communicate within that biofilm. So I'll talk a little bit more about biofilms as we go forward, but I just want to give you that kind of visual. And it's really, really difficult, as you can imagine, because those biofilms are often polymicrobial, meaning it's not just one organism, there might be a handful. And so it's very difficult, if not impossible, to treat because it makes it difficult to get antibiotics or other drugs down into that biofilm. And likewise, difficult to get certain disinfectants and other treatments into that area. Just a second, I've lost my screen. But there we go. Okay. So 60% of human infections arise from biofilm formation. And while most of those are occurring in wounds, they also occur in medical devices and surfaces. And biofilms have not just popped up. They have been around a long time. In fact, it's what forms on your teeth from different streptococcus species. It can form on rocks, like in streams and wet environments. It can form in pipes, within sinks, autoclaves, perhaps on the outside, or in air conditioning vents and things like that that are wet. And so they act as a reservoir, almost a vector reservoir within a healthcare setting, as well as other settings, and persist there over time. That structure I mentioned, that EPS structure, really gives a shield, a protected against antibiotics and disinfectants and other agents, very much stealth, very much off the radar, unless you have a way to detect it, which we'll talk about later. So we typically look at biofilms in kind of that wet or damp environment, right? So catheters, different tubes, IVs, or devices like a knee replacement or something like that. So anywhere you can have a surface, you can have an entrenched biofilm, which is full of microbial pathogens. Initially, they attach. Sometimes it's dependent on the nutrient availability and maybe the temperature. And once you get up to 10 to the 5th, 10 to the 6th, 100,000 colony forming units, you typically have now reached a very strong biofilm that can be very resistant. And this image here is kind of a simplified look at it, but it's a nice cartoon and seeing how they're typically platonic. So they're typically floating around in a perhaps wet environment. And then they can attach via different bacterial organelles, the pili and other things, the cilia, but they can also detach at first. But over time, they will be irreversible and grow into a mature biofilm. And even from that point, they can seed with persister cells away from themselves, almost sending like spies out of that trench warfare, if you will. And here's kind of the scary part of biofilms is that typically when you have a bacterial agent living in this biofilm, it can exhibit anywhere from 10 to 1,000-fold resistance to different drugs compared to just normal bacteria in kind of a wet environment. In fact, in some studies, including the one we show here, looking at Staphylococcus epidermidis, which is a common bacteria on your skin, but can show up in different places and be very dangerous in a non-sterile site. If you treated those within a platonic kind of situation where they're not in a biofilm, 100% of the isolates were usually killed or susceptible at least to the antibiotic vancomycin. But once you put that same bacteria into a biofilm, almost 3/4 of them were completely resistant to the same antibiotic. So again, that biofilm really provides a shield, if you will, to attacking it. Nice cartoon. I've used this in different articles and presentations, and not to get too deep into the weeds here, but you can see that the way pathogens can increase, this is a large bacterial cell, this kind of rod-shaped feature showing you the green stringy DNA. Excuse me. And then over to the right, you see another bacterium. And what they can do is they can really transfer resistance through different genetic mechanisms called mutation events, transformation, conjugation, and transduction. And this conjugation one is probably the most common, where they connect via a pili and they unfold resistance genes through plasmids, these little tiny circular red structures, into the other bacteria. Now, what makes this so dangerous and effective for bacteria is that when they touch, if you can envision using your two fingers touching, it would mean that if I touched fingers with another person, I could transfer a gene to that person horizontally. And that's what bacteria can do. Humans can't do that. Otherwise, I could, touch someone and give them gray hair like I have. Bacteria can do that. And unfortunately, they transfer resistance to antibiotics, antivirals, create, fertility factors and other issues that they can transfer. So all of these are problematic, as this kind of ongoing cartoon shows, creating persister cells and resistor cells and all sorts of issues that bacteria can then share across the environment. And just kind of put this image in your head is if you were on an IV or a urinary catheter and it had a biofilm and it hadn't been changed and you had an issue with a biofilm there, even if you were treating that patient with a high level of IV antibiotics and that biofilm was there, you might kill a certain percentage of bacteria right at the site or even in the bloodstream for a while. But if you don't remove that catheter, or if you don't get a way to disinfect surfaces, it could continually seed resistant cells and persister cells throughout that body so that that infection would re-engage. So it's really problematic if you don't take care of this. They do it through sensing. Bacteria can talk to each other. You may say, how do they do that? They do it through chemical signaling. So they can literally tell different populations, as you see in this cartoon here. It's almost like using a smartphone, but it's a chemical signal to tell other populations to flee, to eat, to go to sleep or become dormant, to multiply, to send out biofilms, and probably a bunch of stuff we're still learning within the research. So again, this makes it very problematic. When you get into dry surface biofilms, one of the big issues is that it's a little more difficult sometimes to sample. So you might have to do destructive sampling. And so you find these everywhere, as this study by Hugh et al. In 2015, and there's many other studies now if you dig into the literature. This one had a very high percentage of finding dry surface biofilms on everything from privacy curtains to doors to mattresses to note clips. And all of those were found even after 2 rounds of terminal cleaning with a 500 parts per million free chlorine solution. So very much embedded into certain types of surfaces and niches. These types of biofilms are prevalent in the hospital environment. They're way more resistant to biocides than those kind of platonic cells we mentioned. A lot of the current cleaning procedures may not be sufficient to remove them. It's absolutely a reservoir for MDROs. For a microbiologist like myself, it can be difficult to culture if you don't know how to sample that surface, which could mean you're looking at a false sense of security if you can't get them scraped up and cultured correctly. And so it's a significant factor in like endemic outbreaks within hospitals. It's absolutely transferable from healthcare workers to patients via their hands or other vector-based types of transfers. Candida, just throwing Candida in here, as a kind of emerging problem. This is a fungal agent. And one of the problems with Candida is not only does it live within biofilms, but it's a eukaryotic organism, meaning it's just like you and I. And there are even fewer antifungal drugs. And so it's really important to prevent these types of infections. You can see cleaning surfaces 2 or 3 times daily can be somewhat effective, but it's just really something to keep your eye on with respect to making sure you have the right disinfectant and so forth. Okay, so this is kind of a little bit of a transition for the next few minutes before I hand it off to Mark, and just kind of showing you some of the technology. So the idea is here, how do you find this surface contamination? And then, what do we do about it? And those are kind of 2 different topics, but first you got to see the enemy. And so currently these are the most widespread methods. Just simply visual inspection might be useful at times. Microbial culture, growing the organism, fluorescent markers, which is putting different types of fluorescent dyes in the environment, and then ATP, which I'll briefly mention here in a second, through a bioluminescence type of detection. Regardless, these typically can indicate somewhat of an overall cleanliness, but not necessarily contamination removal. So those are the 4 primary technology, and I'll just bounce through these quickly before I hand it off to Mark. This is in my wheelhouse quite often, and that's microbial culturing. So the kind of the gold standard for decades was, you go into a place, an environment, you swab it, you might scratch the surface in different ways, and you go through a particular microbiological transfer into a medium, whether it's an agar surface like on this plate you see, or in a liquid form, and you grow it up. And then you look for what it is. Can you identify the bug or bugs, and what types of perhaps antibiotics might be useful against it, or disinfectants? It's a little timely. It can take, anywhere from a few hours to a day or 2. It's gotten better because of molecular techniques in some capacity, but there are some limitations, including, do you have the right type of laboratory to do this? Not all hospitals have that capability. Fluorescent marking, this is using certain substances, commercial substances like gel, powders, and lotions. They glow under a UV light, and so you can kind of mark high-touch objects for cleanliness monitoring. So the idea is that you can visually identify areas easier that may have been missed during cleaning. ATP testing, this stands for adenosine triphosphate. This is absolutely a key organic material that almost all living things have. It's really the powerhouse of your cell. It provides energy to most cells, but it's also a good marker for detecting organic matter on surfaces using bioluminescence, and that helps indicate cleanliness. So, you can use this not only in hospitals, but sometimes food processing areas or other types of areas to identify things that might need re-cleaning, or a way to deal with your protocol, maybe improving it. It basically measures ATP in all living cells left on the surfaces post-cleaning. So high ATP levels indicate poor cleaning. You want very low levels. One of the limitations here is that dead cells may also give off ATP, so that can be somewhat of a limitation. And then you got some of your newer generational things called surface imaging, biofilm, and bacteria scanners. These are typically handheld tablets or lamps. They provide a quick visual kind of measurement of cleanliness and potential risk on the surfaces. You have to kind of look at the color intensities based on signal strength to figure out organic contamination, but it can help in monitoring different types of cleaning thoroughness on hard, non-porous surfaces. And like with anything, as I mentioned earlier, this is all part of a multimodal risk reduction strategy. And Mark will get into some of this a little more, but there are some challenges in the current technologies and methodologies, whether you're talking about microbial testing or fluorescent marking. I'll mention again with microbial testing, there is some instance sometimes with surveillance limitation. For example, some of these bugs you pick up in the environment may not be typical biological microorganisms that you see often in a patient. So there may not be the correct laboratory media or the correct laboratory resources to grow up certain types of things. So there are some issues there. Fluorescent markers need to be applied before cleaning and then examined afterwards to check for removal. So again, it's all about the methodology and following certain procedures. We mentioned ATP. That's can be pretty complex and not always easy to interpret in the field setting. So you really want highly trained and educated individuals that know what they're doing when they're using that testing and its limitations. And then surface imaging testing, right? Kind of making sure you know how to interpret that nuanced understanding of those different types of imaging processes. So I am going to hand it off to Mark now, who will take you a little bit further into the technology.
Rodney, thank you so much. That was a great presentation as always. And next we have Mark.
Okay, can you hear me?
Yep, we got you.
Perfect.
Your presentation is just pulling up. There we go. We see some comments. I think you might want to.
I always do that to see if anybody's
There
paying
we go.
attention.
Hey, there
I
we
always
go.
make sure you guys are paying attention, just when I do that. So, I really want to thank everybody for welcoming me to this webinar. It was thank you to Rodney. He was, fantastic speaker about biofilms. I'm sure we've all learned a lot. He's a technical he's a very technical person. I'm one of those cleaning professionals that really concentrates on the cleaner, the janitor, the custodian, the environmental services person. I've made a career of it. I've like Dan said, I've been here for well over 18 years in all different cleaning and maintenance avenues, and really specializing on cleaning and disinfection and really kind of spreading the word with regards to cleaning and disinfection. So, when we get into it, I'll be a little bit different. It's, how do we really see, what we're doing as cleaning professionals? How do we really get in there? Because, when I look at environmental services personnel or custodial, realistically, they're the first line, or they're one of the first lines of the defense against, bacteria and viruses as we spread through. So, how do we help them? How do we help them become better at their positions? How do we, help them become better at adhering to the SOPs of a hospital or a long-term care facility, those types of things. And really that comes, technology kind of comes into that. Whether it's ATP monitors or visual inspections, fluorescent marking, microbial testing, or this new technology or newer technology called surface imaging. How do we really bring the knowledge at all levels in an infection control environment? And that's really where surface imaging has come in, and that's kind of where it's come about. A few decades ago, we wanted to make things glow. We wanted to make things show up on a surface. So, before, all we did was, does it look clean? Does it smell clean? If it did those two things, we would kind of go along. But as we've just learned, with biofilms, it's a very, very difficult thing, and it's an emerging threat to our existence, really, in healthcare settings, on how to properly remove those things, or how to properly find them is one of the problems. So, when we look at certain inspections, so visual inspection, does it smell clean? This was years and years. Obviously, this is the way we clean. That's how Mom taught me how to clean. Then we've graduated to something called ATP monitoring, or adenosine triphosphate monitoring. A lot of you on this call have seen these monitors. They give you a number on a screen, and interpretation of that number can be a little suspect sometimes. Sometimes you get a guideline, sometimes you don't. Surface imaging is, again, when we look at that surface imaging for the exact same screen, is it clean? No matter what level of cleaning knowledge you have, or infection control knowledge you have, if you understand the color mapping on that image, you can understand, is it bad or is it good, right? So, we wanted to really, and what we really want to promote is color to contamination. Let's bring the visual. Let's spatially make it glow in the eye of the cleaner. So surface imaging has really come along from, there's a few different types of surface imaging technologies. You have certain ones that use a lot of UV light. All of them really use some form of a UV light. There are some that will blast a UV light at a surface, and different frequencies of UV light to essentially make bacteria grow on that surface, and it'll give you a color hue from blues and greens and that type of thing, to other surface imaging where you would have a proprietary spray or a specific tracker that goes on onto a surface, and essentially that illuminates inside of a software technology, or it could be a tablet, could be a phone, so on and so forth. They use UV light to activate those things. So in surface imaging, you get all different things. You either get a glow of a hue of colored light, or you would get your technology capturing that color light. And one of the important things with regards to that is, it'll always come back to training with regards to the cleaning environment. Colors on a surface, no matter what language we speak, no matter what kind of training we have, we can a lot of times recognize those colors. And with surface imaging as well, these can all be loaded within a software-based program, so you can track the results as you go through, which I'll talk about a little bit here. So novel technology, it's specific to bacteria, which is important. So this is specific to bacteria. Doesn't do a great job on viruses, those types of things yet. So there are some drawbacks with surface imaging, but it's definitely a really neat technology to really take somebody through to train. So some of the results with this surface imaging on the screen, especially with computer, a lot of it'll use your yellows, your oranges, your reds, your deep reds for severe contamination. These are all layered contamination with specific sprays. And really, when we look at how imaging works, and a lot of times when we look at different technologies in the market, one of the main benefits of having a tablet base or having a technology base on a surface, we could have the camera exceptionally still, so we could utilize the UV light and the illuminator spray. So on one different technology, you essentially spray the light on a surface. It'll glow it'll glow with a little bit of contamination. On this one, we can actually set up a camera, have all the surfaces, all the high touch surfaces, whatever is in our essentially our audit, our assessment, loaded in the camera. We would spray we would take a contextual image, and all this does is takes a series of 10 pictures on a surface, and then it'll ask you, everything's prompted and everything's instructed. So the camera will then instruct you to spray the spray. Spray the spray. This is essentially a marking spray. It binds to bacteria, fungi left behind, and then it'll glow in essentially the eye of the camera. So the camera will then take another series of 10 images, filter it out, and process that image and automatically upload into the cloud on the system. So really what this allows you to do is track every single picture you do throughout your facility in your quality management software program, moving into the future, so you can pull those pictures at any given time and study those pictures. And essentially each picture would take about 2 minutes to take. So from ATP monitors, when we used to use ATP, well, we still use ATP, but when we use ATP, to set up ATP and to utilize the monitor, it'd take about anywhere from about a minute and a half. Surface imaging is about 2 minutes. And again, when we empower precision cleaning for people, and process it, it all comes down to processes. Cleaning, I always, when I teach cleaning, it always comes down to that process. It's the marriage of chemistry into a process. So we have to have obviously the right chemistry, the right label claims from the CDC guidelines or Health Canada guidelines, depending on which side of the border you're on. But we have to have the processes. Cleaning is not the same as disinfection. It's two separate things. And it wasn't so long ago that the CDC actually released their guidelines for environmental infection control. And in that guideline in 2003, they recommended that hospitals clean and disinfect all their high-touch surfaces, which is kind of obvious to us now. It's proof on the paper kind of thing. And then after that, they really strongly recommended that hospitals monitor their cleaning performance. And that's kind of where we've been stuck in our industry. We've always cleaned, a lot of kill claims, a lot of different technologies of disinfectant, that type of thing, but we've always kind of got stuck in that monitoring aspect. And the reason why we've gotten stuck, in a lot of cases was because it wasn't easy. So what we like to teach is what gets measured gets done, and what's easy gets implemented. And that's extremely important for the environmental services staff, whether you're in a hospital or long-term care, or even in a high school. Monitoring is extremely important. We saw that with the onset of COVID-19. The public has a watching eye on our industry now. It's very, very paramount. So monitoring is extremely important. So one of the powerful tools with regards to surface monitoring or surface imaging is we can show pre-clean and post-clean what that and how it means, how our cleaners are actually doing, how our infection control policies are doing, protocols are doing, so on and so forth. We can show you the contamination left behind. We can show you certain things like films on a surface. We can show you any of the contamination that's not stripped away. And then we can implement that training protocol to remove those properly. So once you're over the surface imaging, surface imaging technology, so once you really adopt that, it's all fine and good to take those pictures. But it's really, really important that you can really go in and say, how do I utilize this data today? But how do I utilize this data in 2 years? How do we compare all of the assessments we've always done? So one of the drawbacks when you look at fluorescent marking or ATP, mainly fluorescent marking, a lot of times when we talk to customers, they say we do paper audits, we'll do Excel-based audits or something like that. And then they go onto a hard drive, or they go into a binder on the shelf. And really, when we're called upon, we scramble to make a report for those things. So, when we looked at their total quality management system on really how do they organize their SOPs, how do they organize their cleaning protocols, those types of things, it wasn't easy, it wasn't fluid. So we bring in the importance of something called a quality management software tool into the total quality management system. And the QMS software is essentially, it's just an organizing software for all of these cleaning surface images or ATP or whichever it's going to be, that you can pull on and call upon at when you need it. Because you can imagine in March of 2020, when we were called upon to, everybody was concerned with COVID. They didn't know what it was. Going into July, August, so on and so forth. When our managers come to us and say, "What are our cleaning protocols? What are our cleaning assessments and audits look like?" There was a lot of paper. There was a lot of paper searching, not as much online searching. So that's where the importance of these QMS systems come in. We really match your SOPs, and we put your SOPs into a quality management software tool with whichever surface assessment tool you want to use. That can be loaded in, and then essentially what it does, it boosts your hospital quality by means of measuring on a consistent basis for the for doing assessments. So you're really looking at continuous improvement because you're constantly capturing data on a regular basis, on a daily basis, essentially. Now, how the QMS improves your total quality management practices. Obviously, when we look at standardization of your procedures, consistency of your procedures, you want to minimize the risk of your errors and your inconsistencies, of course. A lot of these can lead to infection control outbreaks. Outbreaks are, of course, everybody on this call knows, they're extremely bad. They can be extremely costly. They can be costly in dollars. They can be costly in lives. So we really want to standardize our quality management system to the point where we can capture all that data, we can organize it, and then we can train on it. We want to look at that continuous improvement protocol. Continuous improvement, when we capture that data, how do we actually use it? Are we just going through the motions? Are we a supervisor going through the motions? Or are we actually capturing it to say, okay, what are we doing well? What are we doing wrong? It could be as trivial as that. How does this measure up to our infection control strategies for our stakeholders? How do we give all this information for our regulators? Whether we're going through accreditation or whether we have a legal issue, so on and so forth. How do we utilize this information for those types of things? Risk management. We touched on that a little bit. We want to proactively identify these and mitigate these infection risks. We have to say, okay, we understand there's going to be infections, but how do we cut them off before they become a huge problem? How do we put our cleaning and disinfection protocols within the SOPs of infection control, so that when somebody new starts today, they're on the same cleaning protocols as somebody with 30 years of experience? How do we get them up to speed very, very quickly? Performance measurement, continuous performance with regards to environmental services staff going through. How do we and for hospital and long-term care facility points? How are they doing? How are they doing on a regular basis for their infection control protocols? My favorite, training and staff competence. When we go through and we talk to staff on a regular basis, one of the biggest time-consuming parts of any assessment implementation is bringing everybody up to standard with regards to what surface assessments we're doing, and what cleaning and disinfection protocols we're using. And what this quality management system allows us to do is organize all of those SOPs into one area that we can train very, very quickly, and we can uncover any stop gaps in training so that we can improve, and we can score our employees in a positive manner to make them the best cleaners possible. And that's really where the feedback integration comes in, not only from the cleaners, but from the CEOs, presidents, whoever is a stakeholder in this cleaning environment. How do we close that feedback loop for all of the stakeholders, including outside folks that are concerned with the cleanliness of our facilities? And realistically, you want to choose a QMS system that's just dead easy. You want something that has real-time monitoring. So as we're going through doing assessments, it automatically captures everything. You want to be able to take your existing SOPs and digitize those very, very quickly. And you want to have reporting. You want to have reports at the click of a mouse for all of your stakeholders in that, for the powers that be that want those reports. But most of all, it's got to be absolutely simple to use, because we know if it's simple, then it gets utilized. It can't take any more time. It has to improve efficiency. And that's where quality management integration has come. 10 years ago, it was very painful. This day and age, it's becoming just second nature. We can do it with existing devices that we currently have, or even investing in something as simple as an iPad. So digitize your current SOPs in infection control and actually bring them into your place of business. And the benefits of those QMS protocols, we always give insights, and we always give predictive maintenance measures. So if you have a QMS as well as a surface assessment tool, you can see your improvements over time in space specific assessments. So you go through, you can monitor, click of a mouse, have the report ready, and have full training and full assessments going through there. Okay. When you look at a QMS system, you really want to look for partners. You want to look for third-party certification. So one of the main things that we really look towards is securitization of our customers' data, of our patient data, and that's where you want to see SOC 2 type compliance. So they securitize your data. So if you're using any cloud-based software, cloud-based QMS system, you need to make sure that it is absolutely secure, because you obviously, whether you're in Canada, the United States, England, wherever you are, you can't have any data breaches. And then we always recommend partnering with high-level certification bodies like GBAC, which is the Global Biorisk Advisory Council. And what these folks do, they really certify a high set of standards for cleanliness and operational best practices for your biorisk management. And this has been extremely important since COVID. A lot of people want to see this badge on the door of your facility when you walk in. Extremely important to focus on preparedness. It goes through, it helps instruct and it helps guide, develop those SOPs in response to biological threats and risks. And of course, a part of the certification, they want you to have a specific validation. So they want to validate your facility's cleanliness protocols, cleaning protocols, so on and so forth. And then we come into the benefits of assessments and regular audits. And a lot of times our customers will come to us, or we'll see in the main area a lot of regular assessments and audits. Sometimes they're done. Sometimes they can be simple as a four question audit. Is the foyer clean? Is the hallway clean? Is the washroom clean? Yes or no? At least that's a start. Really, what we want to see, we want to have a system that we can build these audits into the future, to the point where you'll start to develop early detection compliance. You want to really drive that continuous improvement with not only your stakeholders, and being completely transparent to all your stakeholders in your company. You want your cleaners to really be enhanced. You want to bring them up because they have a tough job. Asset preservation is a major issue, especially since COVID, with very aggressive disinfectants. With regular assessments and audits, you can start seeing asset preservation by means of, do we need to clean this area as much as we once thought? What is the frequency? What are the processes? Can we change our process? Because of these surface assessment tools, we can now see how clean they are on a regular basis, and we can continuously test to start tweaking our processes moving forward. Regulatory compliance and outbreak management assessments will always show if government agencies come in and you're in outbreak mode and they want to see your reports, you're auditing your assessments, a good quality QMS system is just at the click of a mouse. Again, you don't have to go digging for those papers and procedures and things like that. And of course, with these QMS systems, you want digital tracking and reporting. You want to see your pictures, especially if you're adopting those QMS systems. If you're on fluorescent marking, you want to load those in your QMS. If you're doing microbial assays, you want to load those in your QMS. Whatever surface assessment tool you want to use, you need to be able to load those in, and you want to be able to trend that data, because again, it's so important to have it at a click of a mouse to identify those areas of improvement and importance, and inevitably over time cost savings. Cost savings come in the form of process improvements, asset preservation, choosing the proper cleaners, disinfectants for your building. A lot of times there's no one size fits all. What essentially technology do I have to use to maximize my effectiveness of my cleaning and disinfection program? realistically, for the summary and key takeaways of this whole thing, it was, the talk about biofilms. Biofilms are emerging. It's a very, very important thing to pay attention to. We need to know surface contamination impacts on hospital-acquired infections. This is a major problem and is not going away. And really what surface imaging and QMS systems really do is help you really hone in on your cleaning and disinfection program and on the theory behind it, as well as the real world takeaways with your environmental services staff to go in and really teach them how to clean and why we do what we do. With advanced surface testing and imaging, we can really improve what we're seeing. Doesn't matter what language we're speaking. If we can see that picture, it's really worth 1,000 words. That picture is that spatially, you can see the spatial contamination right on that picture. And really, the bottom line is we have to improve our effectiveness of their cleaning and validation with that QMS. That QMS is really going to organize all that data. And that's really been that missing piece from years ago when we're using fluorescent marking, glow germ creams to ATP and kind of so on and so forth. So, and that's it for me. So if any of you folks are interested in learning about our technologies, please look us up on the web, take the info down, and we can kind of go from there.
Mark, thank you so much. That was a really good presentation. It's good to have Rodney go into the clinical side, and then you're really on the application side to see kind of how we tackle the issues. We've got a couple questions, and the speakers want to go on camera so we can see your smiling faces. A couple questions came in. How does the surface imaging technology work at a technical level, and what kind of data does it provide? And there was also a question, specific bacteria, are you able to track, are you able to look for MRSA or C. diff, and kind of how technical in terms of what you're looking for, I guess, for Mark here?
Yeah, I can take that. That's not a problem at all. Thanks, Dan. So, when we look at what from a technical level, what exactly does the data provide? So in some surface imaging, so what you saw with the camera surface imaging, if they utilize a spray, that spray is it's essentially a binding spray. So it's going to bond to any double-strand DNA, whether that's from fungi or bacteria, and really essentially make that glow in the eye of the camera. The software from the camera, it really layers that contamination on the surface and makes it glow, essentially in the eye, in the picture. It's
Okay.
easy as that.
And there was another question. It's maybe similar, but from Amanda. Can you please share about the technology behind the surface imaging? It's maybe you've kind of answered it. How does the technology identify the bacteria?
So it doesn't identify any specific bacteria. It doesn't, and one of the things that especially is important to note, it doesn't show if it's dead or alive either. So we're not pathogen specific just yet. It'd be great once that can come, but it's not there quite yet. But it just shows contamination left behind essentially after the cleaning step.
And is it just bacteria?
Yeah. So it can bind to bacteria and fungi essentially.
Yeah. Okay. How quickly can the technology provide results, and how does this speed compare to ATP?
Yeah, so again, we touched on that a little bit. It takes about 2 minutes from setup to picture, complete process and storage. So it's about 2 minutes, whereas ATP, if you're taking, it's about a minute with regards to swabbing, processing, so on and so forth.
Yep. And I had a question, like, really, how does a hospital operationalize this? Do they use it for certain thermal cleaning, or what have you shown? And then do they use it along with ATP? If they're already using ATP, do they use that complementary, or how have you seen at this point, how's these technologies used?
Yeah, that's a great question. It's a lot of times when we implement these technologies, because it is a fairly new technology, a lot of our customers are already used to ATP, so they want to still use ATP just as a kind of a fail-safe. just until, I don't know if you want to build trust or whichever. But you can load ATP in our QMS software. ATP can be loaded in along with the surface imaging technology. So you can get a double surface test. So you can see the correlation between both, which is
Yep.
very, very neat until they completely adopt the surface testing.
Okay.
And we use it, and we use it in a lot in environmental services. You can use it in, whether it's operating rooms, terminal cleaning, that type of a thing. Once that spray goes on, you just want to wipe the spray off. But it's immediately after the clean, just like ATP, how you want to test it. And that's going to give you your best results.
And then there was another question, maybe along the same lines. Well, I think on that question, someone asked, what is the spray? I guess they're asking what's in it, or maybe.
Yeah. So again, one of the
maybe secret sauce, but
yeah,
I mean, in general, like general terms. Yeah.
Yeah. It's essentially, it's a biomarker spray that will bind to the, the DNA on that surface. So yeah, it's gonna glow in the eye of the camera. But yeah, there's a little bit of a secret sauce there that we took some time to come up with.
And there was a question, I think you kind of covered this, but maybe kind of nailing down, what are the two major differences between ATP and image tech from a perspective of results?
Yeah, so that's ATP. ATP is great. It's fantastic. It's very limited in the size of the area that you test with regards to ATP. ATP is great on bacteria. It's going to show you the growth of bacteria essentially as it goes through. Whereas with regards to surface assessment, it's going to show. An ATP is going to show you just an average over that small area that you have. Whereas the benefit of surface assessment pictures or colors is going to show you exactly where the contamination on that surface is. So it allows the environmental services staff to actually see. If it's a keyhole, they'll see within the keyhole. Whereas if I test the whole doorknob with an ATP, I may just get the weighted average around the doorknob, and I may not be able to see the problem area. So surface imaging shows exactly where the contamination is. Okay. It's like a guided missile.
There was a question about availability of the presentation. It'll be available at the TIPS website the next couple of days, and you'll get a follow-up email from TIPS, and it'll provide a link. Where's the system being used in Canada, and then how many years has this system been used? And I think in general, I guess.
Yeah, in general. So we had a proof of concept about 8 years ago. It was very cumbersome. It was a little big, but so now it's being used in some large healthcare settings. It's also being used in a lot of post-secondary education, believe it or not. Post-secondary education they're taking their cleaning and disinfection program very, very seriously. Students are watching. Parents are watching, so on and so forth. So this is extremely, extremely important for those folks as well. So we've really seen adoption from healthcare settings, long-term care, as well as post-secondary education.
And there's a question there for Rodney. I don't know if you can see that. What is your experience with biofilm and contamination in healthcare, and especially with floors?
Yeah, so just one quick comment before I answer that Mark just mentioned. I've done so many studies on so many different agents in non-healthcare environments. So as Mark knows, and many people know, there are other problematic areas, including dormitories and recreation centers and physical therapy treatment rooms and prisons. I mean, I've looked at all of these areas personally in my research. So, healthcare is the overarching problem because you think about immunocompromised patients in those areas, but it's everywhere. And so it's not just a problem for healthcare. With respect to biofilms, and could you mention that question again real quick, Dan? I'm sorry.
What is your experience
I see it.
with biofilm and contamination on floors in healthcare?
Yeah, so any surface that is available to be adhered to by a living organism is susceptible to biofilm. It could be a floor, it could be a bed rail, it could be a toilet surface, it could be a dialysis clinic pole for hanging your IV. I mean, just really any place that has a surface is going to be compatible. And I often tell this even in basic microbiology, you know, bacteria especially are notorious for finding a way to survive. They've been found on the highest peaks and mountains and the lowest depths of oceans. They do find a way to survive. So biofilms are pretty much
Survivors.
anywhere. Yep.
Well, we're at the hour. We promised we wouldn't keep you past an hour. We could probably talk another hour, but Rodney and Mark, thanks so much for your great presentations. Again, as I mentioned, there will be a copy on the TIPS website. We also recorded this presentation, so there'll be a recording. If you were not able to join, you'll be able to view the presentation, and then stay tuned for the next webinar with TIPS, masterseries.events. Everyone have a great day.
Thanks, folks.